Hi, my name is Clementine Brannigan, and this summer I was lucky enough to participate in an 8-week summer research project at the James Hutton Institute, funded by BSPP and supervised by Lorena Ronhal. My project looked at how the surfaces of barley leaves affected the microorganisms living on the phyllosphere. Leaves have an epicuticular wax coating made up of various long-chain fatty acids, the exact composition of which varies between species as well as across developmental stages. As the first barrier to host colonization, as well as a habitat for many microbes, this wax layer has the potential to impact the diversity of species found in the phyllosphere, as well as the severity of the infection that they cause. Therefore, my project aimed to characterize the bacterial and fungal communities living on the surface of barley leaves with various mutations. We grew multiple mutant lines of 2 barley variants and collected leaves for sampling at 2 time points through the growing season. These leaves were frozen and ground in liquid nitrogen, then DNA was extracted. Much of this project was consumed by conducting a series of PCRs to find the best way to maximize the ratio of microbial DNA to plant DNA in the samples. This was so that I could prep a library for Illumina next-gen sequencing to be used for identification of the variety of species hosted by the barley. I also ran qPCRs to compare the biomass of the barley scald pathogen, Rhynchosporium commune, across mutants from the same parental line. This year had fairly little disease, particularly early in the growing season. We did not find any significant differences in relative fungal biomass between lines. Finally, I established a culture collection of yeasts, bacteria, and fungi found on the leaves of a wild-type barley variant, Diablo. I spread leaf wash on LB and PDA plates, then picked colonies to isolate and store. The goal was to capture the full range of diversity found in our samples. Here, you can see me prepping fungi for long-term storage. Overall, this project was a really great opportunity for me to develop additional lab skills, especially working in sterile conditions. I also built soft skills such as problem solving and communication. I'm really excited to see how I can build on this experience and continue my career into the important field of plant pathology research. Thank you very much to BSPP for funding this project, to JHI for hosting me, and to Lorena for her supervision.